Gabrielle Black. Gabby is a postdoc scholar at the Civil and Environmental Engineering Department in the University of California, Davis. Gabby is currently investigating chemicals in drinking water and potential correlations to historic breast cancer occurrence rates in California, in addition to evaluating pyrethroid pesticides and PFAS in different environmental compartments. Prior to her postdoc, Gabby earned her Bachelor of Science in Molecular Biology at the Dominican University of California and spent 2 years as project manager for a non-profit and an affiliate researcher at Lawrence Berkeley National Laboratory, investigating the microbial community influence of aerobic human waste composting in Haiti. Gabby received a PhD with Dr. Thomas Young in Agriculture and Environmental Chemistry at the University of California, Davis in 2019. So thank you so much for joining us here. How did the weather go last night? Didn't get flooded out?
No, we were good, and we're learning today of how bad everybody got hit. So I think the San Francisco Bay Area got 11 inches, which I should have done the conversion to centimeters. I'm sorry, but it was a lot.
Rightio.
We're drying out.
Oh, that's good. I'm glad to hear. We can see the presentation and we can hear you fine.
Okay, great. Well, thank you all for joining us, and thank you, Brad, for the invitation to speak. I am currently a postdoc at UC Davis working with Tom Young, and today I'm going to talk about a project that's been pretty near and dear to my heart for the last 5 or 6 years. But I did this largely in part as part of my PhD dissertation. And so the topic was looking for estrogenic consumer product chemicals in California sewage sludge. We did this with a combination of non-targeted chemical analysis with a suite of in vitro and in vivo estrogenic measurements. Oops, why isn't that advancing? There we go. To do this work, we partnered with the California Association of Sanitation Agencies, and they put us in touch with 12 wastewater treatment plants in California that were willing to contribute some excess sewage sludge for our research. We had 2 utilities that went above and beyond. They were our overachievers that sent us 2 samples. So these 2 wastewater treatment plants produced 2 finished product sludges, and under the US EPA federal regulations for sewage sludge, there's Class A and Class B sludges. Class A is the better sludge that's undergone more rigorous treatment versus the Class B sludge. So 13 of these 14 samples were Class B. We had 1 Class A sample. And it was surprisingly difficult to get utilities to hand over sludge when you tell them you're looking for contaminants that no one's regulating yet. We spent a little bit longer than I like to admit establishing a sample prep method for this work. So ultimately we landed on a fractionated pH adjusted sonication extraction with a solid phase extraction cleanup and several washes to get rid of some of the gunk in sewage sludge that causes instrumental issues. We ultimately ran these extracts on an Agilent 6530 LC-QTOF, and we acquired all of our data in what's called data independent acquisition. So what's nice about this is that we get a lot of information about compounds present without having to know what those compounds are ahead of time. In addition to that, we get the molecule as a whole, in addition to some fragmentation information, which comes in really handy as we move on. So with this data, we're able to analyze it in 3 different ways. So we can first look for the known knowns. So compounds we know are probably in sludge. We can order our pure chemical standard, run a calibration curve, and then quantify that compound in each of the samples. For this work, we didn't really do too much targeted work except for calculating surrogate recoveries and spike recoveries. We really wanted, our goal of this work was to identify compounds that maybe haven't already been identified in sewage sludge and are potential emerging contaminants. In the US, we land apply over 60% of our sludges, and we create something like 60 million metric tons per year. So identifying what compounds are potentially problematic but not currently regulated was our goal. And we didn't know what those were ahead of time, so we didn't want to quantify anything or order a boatload of standards that ultimately resulted in a bunch of non-detects. So the second way we analyze our data are for known unknowns, so compounds that we know are contaminants, but maybe we don't think that they're in sludge. So to do this, we have a library of about 5,000 compounds that have been acquired and curated in a similar acquisition mode that we ran our samples in, so we can do a fairly nice side-by-side comparison. And this allows us to screen through 5,000 or 10,000 compounds at a time in a semi-automated fashion. And then lastly, which I think is the fun part of what we do, is non-targeted analysis. So looking for the unknown unknowns. So these are maybe transformation products of known compounds that no one's measured before. Maybe they're compounds that are metabolites, or maybe they're compounds that just aren't in our spectral libraries. To do this, what's nice about the way we acquire data on our high-res instrument is we can do this somewhat unbiasedly. So any mass that's detected on our mass spec has potential to be identified. So how this works is we can look at that mass at every retention time, and then through a variety of statistical filters, we can prioritize which features are necessarily important for whatever question we're asking. And then we can generate molecular formulas and search open source databases like PubChem or ChemSpider and try and identify and predict how a compound would fragment if it was in fact that fragment in that database. And this ultimately allows us to look for hundreds of thousands of compounds if we can detect it on our mass spec. And for that, we use a suite of vendor software and open source software, and it has a lot of creativity in that data path, which is kind of exciting. But I'm going to save that for the second part of my talk today. So for the first part, we're going to talk about suspect screening and our results there. So to start, we have a library of about 5,000 compounds that have MSMS spectra, and this is a merged library of the Agilent Personal Compound Database libraries of forensic tox, water contaminants, and pesticides. And when we ran our 14 sludge samples through this database, we identified 118 unique compounds. Because we're doing all of this work in the through the lens of estrogenic compounds, we took those 118 compounds and ran them through a quantitative structure activity relationship model. And we downloaded this program called Vega. It's a free download. It's super easy for someone that's not good with coding like me. You download this interface and you input SMILES strings and then identify which toxicity endpoint you want to predict. And within seconds, you have predicted values with an ADI, which is really nice. So for this work, we looked at 3 different endpoints. So the first is the estrogen receptor binding affinity. So how well a compound is predicted to fit within that receptor. So hormones and receptors are kind of like a lock and key. So it has to have a backbone or a ridge that can fit into that lock, which is the estrogen receptor. Second, we looked at estrogen mediated response effect, which looks at if a compound can bind, what's the likelihood that it's going to cause a cellular effect? And then lastly, we looked for compounds that were predicted to be reproductive or developmental toxins. So this work, our overarching goal, again, was to look for compounds in consumer products that can be highly regulated or phased out. We anticipated identifying a lot of pharmaceuticals. Those aren't necessarily what we're trying to focus on because we're not the right kind of doctor to say we shouldn't use those in consumerism. But of the 118 compounds we detected several plasticizers, which kind of overlays nice with Chelsea's talk. Bis-2-ethylhexyl phthalate, or DEHP, is one of the most commonly used plasticizers here in the US, from my understanding. We also see some congeners of that diethyl phthalate. And then we also identified bisphenol A in one of our samples. Like I said, we expected to see a lot of pharmaceuticals and hormones, and that we did. I think it's important to identify them, realize that they're coming, they're persistent through their intended use as an antibiotic or an antihistamine or a hormone, but that they persist through wastewater treatment and have the ability to be an environmental issue later on. And then we also identified a few pesticides, some cannabinoids. Ethyl 4-hydroxybenzoate is ethylparaben, which is a preservative found in a lot of food products and cosmetics. And then benzophenone 1, which is a UV filter. So there are some consumer products in here, consumer product chemicals in here that are persistent through the sludges and are predicted to have an estrogenic effect on the back end. And so our goal is that, maybe we identify that and do some further research and hopefully promote further monitoring in consumer products. So on to the non-targeted work that we performed. So non-targeted is is my favorite part of our research. Like I said, any mass that is detected by our mass spec has the ability to be detected if it falls within several parameters for our research question. So we start off by deconvoluting and aligning all those masses. So this plot on the left, each peak is the same compound or the same mass, the same retention time. And so we end up aligning them and we get this beautiful matrix of mass A at retention time B in sample 1, 2, and 3. So we can do some pretty robust statistical tests. When we do this for our 14 sludge samples, we end up with over 15,000 features. And we could spend a lifetime identifying these 15,000 features, and we would have spent that lifetime identifying about 14,000 compounds that aren't exciting and aren't really all that important. Sludge is really messy stuff. We detect a lot of humic substances and lipids. We need to get that data set down into a more manageable number so that we can try and identify compounds that are potentially estrogenic. So in this case, we did a simple detection frequency filter. So when we look at features that are present in 75% or 10 of the 14 sludge samples, we're able to narrow that down to about 3,000 features, which is a much more manageable number, but still would take decades to identify all of. We then wanted to tie this into the estrogenic piece. So at the time, there were about 40,000 compounds in the norm and suspect exchange of environmental contaminants. We combined those with about 5,000 compounds from those adjuvant libraries that didn't have spectral information associated with them. And we fed those 45,000 compounds through this Vega QSAR model to predict which of these 45,000 compounds can interact with a estrogen receptor. When we did that, we got about 5,000 compounds that were predicted to be estrogenic. We then filtered our 3,000 features through that list of 5,000 compounds. And again, this is just an accurate mass screen. So what's nice about having a high resolution instrument like our LC-QTOF is that we have a very exact mass out to 3 to 4 decimal points. So doing an accurate mass screen is not as good as running a spectral library screen, but we can still have a little bit of confidence in that mass because those masses are so exact. So when we do that, we get about 175 matches. We then take those 175 matches and run targeted MSMS. So one of the constraints of our initial data acquisition mode is that when you have a matrix like sludge that has a very high chemical richness, you get a lot of co-eluting compounds. So you get a little bit of a messy spectrum. So when we do targeted MSMS, we have a nice clean spectrum. From there, we've started to lean on our big brothers of non-target, which are the metabolomics field. And we're starting to use some of their software for molecular formula generation and structural annotation. So with that nice, clean, targeted MSMS spectra, we're able to generate a molecular formula. And then this program, MS Finder, that we've been using will then search open source databases like PubChem, and it'll look for structures with that molecular formula that was generated based on our accurate mass and fragmentation information in that targeted MSMS. That structure is then fragmented in silico, which means it predicts the fragment, how it's going to fragment, and then it'll try and match the fragments it predicted with the fragments observed in our targeted MSMS and score it. We strive for a level 1 or 2 identification, which means that, like in this case, we have estrone at a 6.52 out of 10 score. We then order this or run this compound estrone and see if the compounds elute at the same time, if they fragment at the same time in practice, and that's a level 1 identification. When we ran all 14 of our samples through this workflow, we identified 2 compounds. So another plasticizer, diisobutylphthalate, which is really common in consumer products, especially soft plastics. So this is present in shower curtains, plastic wrap. And I recently heard that phthalates were the source of that delicious new car smell. And if that's the case, I'm really bummed that they haven't made an air freshener that makes your car smell brand new year-round for many years. Although in doing this work, it was a little bit of a bummer to hear that that smells maybe not so good for you. And then our second ID, so these are compounds that are present in over 75% of these samples. Our second ID popped up as levorphanol, which is an opioid pain medication. But in digging into this compound a little bit more, I found a paper that referred to it as the forgotten opioid in the United States because it's been replaced with longer lasting, less dependency medications. And so in doing this, I figured out that levorphanol has an optical isomer called dextrorphan, which is the primary liver metabolite of dextromethorphan, which is the DM in like Robitussin DM or Mucinex DM, the cough medication. So we confirmed levorphanol with a chemical standard, but based on usage statistics, we really feel like it's pretty unlikely that it's levorphanol and that it's probably a dextrorphan. So we're not totally sure how to classify this on the confidence scale for this, but we're just saying it's dextrorphan or levorphanol, but it's probably dextrorphan, which is an interesting debacle that's not really talked about when there's optical isomers. In addition, so like I said earlier, we had these two overachiever wastewater treatment plants that provided 2 samples each. And we kind of were opportunist about this set because we realized that the two samples from each utility were really a before and after additional treatment. So we have this mesophilic treatment set where we have a sample coming out of the wastewater treatment plant that's been held at 35C for 15 days, and it's about to get dumped into this open air digestion pond where it sits for 5 years. And at the same time, or at the time of sampling, they had just dredged out that digestion pond. So we got a kind of after mesophilic treatment sample as well. And then as far as the thermophil treatment goes, the material came out of the wastewater treatment plant as a class B sludge, and then underwent this 2-day hold at 55C followed by a 10-day hold at 40C. And the result there was a class A sludge. So we wanted to look at features that increased and decreased with each of these treatment sets by a factor of 5 or more. And we also correlated that increase and decrease in treatment with measured estrogen receptor agonist and antagonistic activity using some cell-based bioassays. So the bioassays that we're using are a genetically engineered cell, or genetically engineered human ovarian cancer cell. So we grow these cells and adhere them to the bottom of these 96 well plates and then feed our extract onto these cells. And these cells are engineered to be really rich in estrogen receptors. So any compound that mimics an estrogen will bind to that cell. We let that incubate for about 24 hours. And then at the end of that incubation period, we break open the cell and measure activity. And the way these work, any compound that mimics an estrogen, if it binds to that receptor, it elicits this transcription of firefly luciferase. So when fireflies glow, it's actually an enzymatic reaction of luciferase with the protein luciferin, and it creates light as a byproduct. So this cell line has been engineered to have that luciferase gene. So as soon as a hormone mimic or a compound binds that estrogen receptor, it elicits transcription of firefly luciferase. So during that 24-hour incubation period, if there's a compound that's highly active on this receptor, that cell will just be churning out luciferase, and we'll have a ton of it at the end. At the end of this incubation period, we break open the cell so we have free-flowing luciferase, and then we put the plate on a plate reader and add luciferin, and then we're able to quantifiably measure the light that's produced as a byproduct of this luciferase degrading the luciferin. And all of this is measured in relation to a 1 nanomolar estradiol control. So when we ran this on our 4 samples, these are the pre and post mesophilic and pre and post thermophilic sets, we see about a 20% reduction in the mesophilic treatment, but then we see this 85% reduction in the thermophilic treatment. And it was, at first we were thinking like, oh wow, class A sludge is like really good, it's not estrogenic at all. And then we became a little bit suspicious because every other sample had at least a little bit of activity, and then to have one with none was a little fishy. So apparently complex mixtures are complex, and there's often many mechanisms for cellular activity that are fighting against each other. And the 2 main mechanisms are agonism and antagonism. So agonism is when a compound will bind to a receptor and elicit a cellular response, versus antagonism where it binds and inhibits a response. So we wanted to see if we had, in fact, antagonism or antagonistic compounds present in this sample. So we fed one cell, one plate with 1 nanomolar estradiol, and then we fed another cell with 1 nanomolar estradiol plus that thermophilic post-treatment sample. And we saw over an 80% suppression of that estradiol, suggesting or providing evidence that there is in fact a lot of antagonistic behavior or antagonistic compounds present in this sample. So we then ran the antagonistic assay on all 4 of these samples and saw this really strong negative correlation between compounds with high agonism and low antagonism and vice versa. So we then were interested, in what compounds are increasing with increasing antagonism and decreasing with decreasing agonism. And we ultimately found quite a few pharmaceuticals. So everything, potential agonists, so things like metoprolol and fenofibric acid. Oddly, we find a lot of heart medication and cholesterol medication in California, which this is evidence of, but hopefully that doesn't speak too much about the overall health of Californians. And then potential antagonists, we see mestranol, which is one of the active ingredients in birth control, which makes sense because that's the mechanism and the point of birth control is to bind to a receptor and prohibit a response. We found an azo dye and an industrial chemical. But what this really led us to think about is that whole extract activity. And a lot of effects-directed analyses will only look at agonistic activity. And I think what this proves is that it's really important to look at both sides of the coin here, because had we not run the antagonistic assays, we probably would have, we wouldn't have been able to help making some claims about how great Class A biosolids are. Part of the next chapter of my thesis that has bled over into my postdoc has been doing an effects-directed analysis. So in kind of the beginning of this work, we ran the agonism and antagonism bioassays on all 14 of our samples, and we again see a really good negative correlation of that antagonism versus agonism. Our next steps, what we're working on right now, is to perform a correlation with feature abundances and observed bioassay activity. And we have found that this elastic net regression is really a robust tool for doing this correlation. We recently did this and found about 40 features that were correlated with agonism and antagonism. There are 2 features that overlap, which I am feeling optimistic about because one has a positive correlation coefficient and one has a negative. So our next steps here are to perform targeted MS-MS and try and annotate those 40 compounds that are strongly correlated with both endpoints to try and identify kind of the really problematic compounds present in sludge. Part of that effects-directed analysis will include fractionating our chemical extracts, rerunning those fractions on the bioassays, and keep doing that until we can identify single compounds or classes of compounds that are responsible for observed estrogenic activity. And we've started to do this, and we've narrowed down a lot of our activity to this fraction 2 once we fractionated by 5 times of our whole extract. So we just need to keep fractionating fraction 2, I guess, until we get a more clear picture of what's causing that activity. And with that, this has been a highly collaborative project, and just want to thank a lot of people in the lab, our collaborators at Agilent, our contact at CASA, who made all of this work possible by bridging the gap between researchers and utilities, and then a whole host of funders. And so with that, I think there's a couple questions, couple minutes for questions, Brad.